Dear students, to today's science class which deals with animal tissues, part of chapter 6 of NCRT. I am your teacher, Deepika Singh Rajawat. Tissue is a group of cells having same origin and similar function. Same origin and similar function. This is very important that the whole group of cells will be performing the similar type of functions. Next, let us quickly see the types of plant tissues, revise them. They are broadly of two types, the meristematic tissue and the permanent tissue. Meristematic tissues, cells are continuously dividing and giving rise to more cells. So they are responsible for the growth of the plant. Whereas permanent tissues, they stop dividing and they become specialized or differentiated to perform different functions. Well, meristematic tissue, according to their position in the plant, they are of three types. The apical tissue, that means they are at the apex of stem and root. The intercalary, that is they are in between the tissues, and both these are responsible for increasing the length of the plant. And the third type is the lateral tissue, which is responsible for increasing the diameter of the plant or the width of the plant. Coming to permanent tissues, they are again of two types. The first type is the simple tissue. It is a tissue which is made up of only one type of cells, and they are of three different types: the parenchyma, the collenchyma, and the sclerenchyma. I hope you remember their structure, their location, and the function. This becomes very important as a 5-mark question in the theory exam, as well as it is part of the practical also. We will be seeing these slides in the laboratory under the microscope. Then the second category of permanent tissues are the complex permanent tissues. Why complex? Because they are made up of more than two types of cells in the tissue. In this case, there are, if you remember, 4 types of cells. And these complex tissues, they are of two types. The xylem, responsible for transport of water and minerals, and it is made up of 4 types of cells: the xylem parenchyma, the xylem sclerenchyma or fibers, tracheids and vessels. Whereas the second type is phloem, which is responsible for transport of food, and it is also made up of 4 types of cells: the phloem parenchyma, the phloem sclerenchyma, the sieve tube cells, and the companion cells. In addition to that, there are 2 more special type of tissues. One is the stomatal apparatus, made up of stomata, guard cells, and the surrounding epidermal cells. It is responsible for exchange of gases as well as loss of water vapor or transpiration. The second type is the cork cell or cork tissue, which are dead cells. They have got suberin in them, and they are found as outermost tissue. In the plants, they appear as brown color in the bark, on the stems and the branches of older trees. So this was a very quick revision of different type of plant tissues. Now let us go on to the animal tissues. Now, depending upon their origin and the different characteristics, they are divided into four types. The first type is the epithelial tissue, then the connective tissue, the muscular tissue, and the nervous tissue. Each of these we will be seeing in detail in the following class. To begin with, I'll take a very simple tissue which you have some previous knowledge, and that is the nervous tissue. Now, each of these tissue, they have their own characteristic features, but let us go one by one. Nervous tissue is made up of cells which are very elongated. If you remember, nerve cells are supposed to be the longest cells in the human body. They may be as long as 1 meter. Now, because they are elongated, they are also called nerve fibers or neurons. Under the microscope, this is how nerve cells appear. There is the cytoplasm or the cell main part in the center. You can see the dark nucleus, and it has got many processes, big and small. Then let us study the characteristic. The first very important characteristic is the excitability. The nervous tissue is excited on receiving a stimulus. Now, what does this mean? Nervous tissue, if you remember, helps the person to react to the surrounding changes. So, whatever changes that are taking place in the surrounding of an individual will be sensed by these cells. Not every cell in the body is able to sense these changes. Not your skin cells, not your blood cells. It is these nerve cells which are able to sense the change in the surrounding. Say, for example, your eye is a sense organ, part of the nervous system, and you are standing in your balcony and you're looking at the tree in front of you. So the message is going to the brain. The picture is that of the tree formed by the eye, and the brain is analyzing and telling you that it is a tree. The moment you change the direction of your head and you see, for example, a bird in the sky, immediately the environment for the eye has changed. Your eye is now sensing something else. And that picture it is again sending to the brain, which is analyzing and telling you that it is a bird. So this is excitation. So whenever we say it is excited, we say there is a stimulus. And then this stimulus is passed on to other parts of the nervous system like brain and spinal cord through nerves. So this characteristic is called conductivity. It is able to conduct that message or that stimulus that it has received in the form of impulse. When this message travels, we say impulse is generated. So these are two very important characteristics of nervous tissue: excitability and conductivity. Now location, very obvious, all of you know. Nervous tissue forms the brain, the spinal cord, and the nerves. And functions include responding to stimuli. Now let us take one more example. Say, for example, you see a dog coming towards you. That picture is sensed by the eye, quickly transmitted to the brain, where in the memory of the brain, it tells you that this dog is going to bite you. The picture is that of a dog which is snarling towards you, which is angry at you, it is coming quickly towards you. So, the body can give two responses. Either you run from there faster than the dog, or you pick up a stone or a stick and try to scare the dog. So, where is the information going? From the brain, the message will go to the muscle. If you're planning to run, then it is your leg muscle which will give the response. Or if your reaction is that you will stand there and you will try to scare the dog, then the reaction is given by your hand muscle, which will either pick up a stone or a stick which is lying nearby. So these two tissues, they work together. The nervous system or the nervous tissue and the muscle tissue. Now let us look at the simple diagram of a nerve cell, which may be as long as a meter. This is your familiar diagram. You have also drawn this diagram before. This diagram is important from your theory point of view. Very often in the previous years they have asked it for 3 marks, either to draw a label diagram or to they will label the parts as ABC and they will ask you to identify it. Anyway, a neuron consists of short processes, branched simple processes called dendrites. Single is dendron, plural is dendrites. Then there is the familiar nucleus, very important part of the cell, the cell body or cyton. Here is where all the processing is done, where whatever the message you are receiving, that is processed in the cyton of the neuron. Then this long process is axon. It is actually, you can say, it is like a wire, and that is going to transmit the impulse. Then there are at the end what are called the nerve endings, from where this impulse will be passed on to another neuron or to the muscle. So children, I hope this diagram, this simple diagram which is there in your NCERT, is now clear to you, all the different parts and the functions that they perform. The dendrites, they receive the stimulus from the other neuron or from the surrounding. The cell body will be analyzing it, whatever the impulse is, and then the axon will be passing it on. Nerves would be again passing it on, nerve endings would be passing on the impulse to another neuron or to the muscle. Then we come to this picture again from the under the microscope with the label diagram. Now you can see the long axon, the cell body with the dark nucleus in the center, the small process dendrite, and one interesting neuroglia. Neuroglia is not in your syllabus, but some of you like to go for higher references and other reference books. Neuroglia is nothing but, these are simple cells. They don't have the characteristic of nervous tissue. They're only there as packaging cells. They are supporting the neurons surrounding them. They are making them into nerves or into brain or into the spinal cord. They are packaging cells. Then we come to the next type of tissue, the muscle tissue or the muscular tissue. Again, if you see, the cells of this tissue are usually elongated. These are long cells, so they are also called muscle fibers. And their characteristics include, again, excitability, that is, they are able to respond when they receive the signal from the neuron, from the nervous tissue. And then the muscle shows contraction. That is the second characteristic, the contractility. The muscle is able to shorten, it decreases in size, and that is called contraction. So, as it receives the stimulus, it contracts. And when it contracts, it pulls the bone which is attached to it. And that brings about movement. Or when it contracts, say for example in your stomach, then it brings about movement of the stomach. So these are located along with bones in the walls of organs like stomach, intestine, heart, which show contractions and relaxations. They are not there in the kidney and the lungs, for example. Then functions, they help in movement of limbs and other body parts, including the internal organs. Then we come to the type of muscular tissue. Now, depending upon location, they are classified as three types. First is the skeletal muscle. From the name, it is very clear they are found along with bones. Then there are the visceral muscles, which are found in the viscera. Viscera are the internal organs like stomach, intestine. And third are the cardiac muscles. As the name is suggesting, they are found in the walls of the heart. We will see individually all these later on in the class. The second type of classification depends on the structure. So they are either striated, that means they show some bands on their structure. So from these three cells you can see two of them are having these alternating light and dark bands. So these two are also called striated muscles. Whereas the second category are the smooth muscles. As you can see in the visceral muscles, there are no striations. No layers are seen. So they are also called smooth muscles. Let us see each one now individually. But before that, here is an overview, a quick review of the three types of muscles. A very good picture depicting the location of the cardiac muscle in the heart, the skeletal muscle along with the bone, and the smooth muscle in the wall of the stomach. This is for quick revision. Now let us look at the characteristic of skeletal muscle fibers, which are found associated with bones and they help in their movement. Now this is a picture which I have taken from your NCERT, and I have clubbed the three muscle fibers to show this is how they are arranged in a muscle. They are in bundles. So the first characteristic is that they are unbranched cylindrical fibers. Unbranched, as you can see in the diagram, they are not showing any branches. They are cylindrical, means they are solid rod-like structures, and fibers because they are long cells. The second interesting feature is that they are multinucleate. This is a very good example of cell which is having many nuclei. You can see these nuclei are there, and mostly these nuclei are more towards the outer side, that is the periphery. The third characteristic is that they are striated. they show alternative dark and light bands. The reason for that is in detail you don't have to do. That's because of the arrangement of the proteins in them. They are giving the appearance of striations. Then the fourth category, very interesting, fourth characteristic, and that is that they are voluntary. Voluntary means they are in your control. You can move these muscles as your wish. So you can see you can move your hand or you can move your leg according to your wish. And the last characteristic is that they get tired or they get fatigued. Many a times you may have experienced that when you are sitting on a chair for a long time, say when you're studying, when you're revising, your back starts aching. Or when you go for a run, a long distance run, then the muscles of the leg start paining. And that pain is because the muscle is showing the characteristics of fatigue. Now let us see how this muscle looks under the microscope. Beautiful slide I have tried to capture for you. Since we are not yet going to school, but we also should be able to complete the practicals. So one of the practicals is to study these cells under the microscope, these tissues under the microscope. So here I have tried to capture a picture and show you how the muscle tissue looks under the microscope. You can see that they are unbranched cylindrical fibers. Cylinder because they are of uniform diameter throughout. Then you can see the tiny dots, so many of them, indicating the multinucleate condition. Beautiful striations can be seen, alternative light and dark bands. Then they are voluntary cells and then they exhibit fatigue or they show tiredness. Now you must be wondering why it is pink in color. Our muscles are not that pink. It is only because some stain has been used to prepare the slide so that all the characteristics are seen very clearly. Going on to the second type of muscle fiber, that is the visceral muscle fiber. They are found in the walls of organs like stomach and intestine. Here you can see a picture of the stomach wall showing you the visceral muscle fiber, and it's a smooth muscle fiber having spindle shape. Now spindle shape, if you look very carefully, it is swollen in the center and pointed at the ends. Now this is characteristic of a tool which is used by weavers. When they are weaving the cloth, when they are making the cloth, they are using this spindle, and from there this term comes spindle shape. Uninucleate. Only one nucleus is seen in the cell. Again you notice that all these cells are together in a bunch. They are non-striated. In the picture from your textbook, they are showing striations, but you don't draw them. There are no striations. I will show you the slide also. The next characteristic is that they are involuntary. They are not in your control. You cannot control the stomach of your, the muscle of your stomach, And then they never fatigue. They don't exhibit tiredness. This is how it will look under the microscope. See very carefully, you don't see any striations, and they are so tightly packed together that almost they are giving the appearance of multinucleate, but they are not like that. Very carefully, if you notice, they are spindle shaped. Only one nucleus is seen, uninucleate. You don't see any striations. These are involuntary, that means not in your control, and they never fatigue. Let us look at the third type of muscle fiber, which is the cardiac muscle fiber found in the walls of your heart. And this is what it looks like. Again, this diagram or picture is taken from your textbook. You can see that these are branched cylindrical fibers. Skeletal muscle was unbranched. Here, these are branched. Just to show the branching, in the diagram they have shown little gap in the fibers, although they are very close together and the gap is not so clear as it is shown here. Then the second characteristic is that they are uninucleate. You can see in every single cell there is only one nucleus. Then they are showing striations, striated muscles. They are involuntary. You cannot control the muscle of your heart. And they never fatigue. They never get tired. Right from the time the heart develops very early in the embryo and it starts beating, from that time onwards, the heart muscles do not stop working. Can you tell me what will happen when they stop working? It will result in the death of the individual as heart attack or cardiac arrest, whatever. Then let us see how it looks under the microscope. See how closely packed these are. Branching is not very clear, but yes, they are branched cylindrical fibers. Then a very interesting feature is seen. Between the two cells there is a dark line. We call it intercalated disc. It is responsible for quick transmission of the impulse, so that the whole heart is contracting very quickly and together. Then you can see the striations very clearly in the slide. Then the uninucleate condition, only a single nucleus can be seen. Then these are involuntary, they never fatigue, and they always show rhythmic contraction and relaxation throughout life. The heart is beating with a particular rhythm. Now here is a quick description, a quick revision of the three types of muscle tissues. Again, a very important question for 5 marks in your exam. Very quickly, the skeletal muscle fiber. These are unbranched cylindrical fibers showing multinucleate condition. They are striated. They are in your control, that is, they are voluntary, and they show fatigue or they show tiredness. The visceral muscles or the smooth muscle fibers are spindle shaped, uninucleate, non-striated, involuntary, and they never fatigue. Lastly, or the third type of muscle is the cardiac muscle fiber, which are branched cylindrical fibers, uninucleate, striated, involuntary, and they too never fatigue. Now coming to the end of this video, to the end of this class, this is very important both from theory as well as practical point of view. For practicals, I'll be taking a short video discussing these, how to draw them, etc., what to write about them in another video. Now let us quickly revise or recapitulate what we have studied in today's class. In this diagram, you are asked to label the parts A, B, C, D and E. So this is the animal tissue is the neuron or the nerve cell. Be very careful of the spelling neuron, where A is the cell body or the cyton, B is the nucleus, C the dendrites, D is the axon, and E is the nerve endings. Next question, identify the following animal tissues and state their characteristics. The first one, I hope you can identify. It is the cardiac tissue. You can see the branching and the striations and the intercalated disc. Now here B, because of the spindle shape, because of the uninucleate, because of no striations, you can identify it as smooth muscle fiber. Then because of the striations, the multinucleate, no branching, this is skeletal muscle fiber. And lastly, these are the nervous tissue. You can see the many processes, axon and the dendrites in this tissue. Similarly, the same thing can be asked in another way. You are asked to identify the tissue A, B, C. A, because it is found in the along with the bones, it is the striated muscle. Then B, it's found in the wall of the heart, so it's cardiac muscle. And C, they're found in the wall of the intestine. They are the visceral muscles or the smooth muscles. Well, dear students, after studying the muscular tissue and the nervous tissue, we still have some time left to study the practical related with this chapter. I know 2 animal tissues are still remaining, but their content I will not be able to complete in this short time. So at least we can utilize this time and look at the practical which is related with this chapter. So the aim of the practical is to identify parenchyma and sclerenchyma tissues in plants, striated muscle fibers and nerve cells in animals from prepared permanent slides and to draw their labeled diagrams. Now let us see, when we are writing the practical in a copy, it should have foremost the aim. Again, to identify parenchyma and sclerenchyma tissues in plants, striated muscle fibers and nerve cells in animals from prepared permanent slides and to draw their labeled diagrams. Now in this aim, there are 2-3 things which we should be very clear about. First of all, it says permanent slides. Now when you have made the onion peel slide and the human cheek cells, those were temporary slides. They would dry up and we cannot store them. Permanent slide preparation is a long procedure and for higher classes in colleges. So permanent slides are available in the market, in the shops which are selling scientific equipments, which can be purchased and they can be seen in the laboratory under the microscope. Now since this is a virtual practical, what I will do is, I cannot take you to the actual laboratory. I have downloaded these pictures from the internet and we will see how they look here in the slide. Then the second term here is to identify. Now identify on what basis? Obviously the structure. What you are seeing, there has to be some identifying features. You cannot say I am seeing dots or I am seeing some circles, not like that. There will be at least 3-4 identifying features that will be specific to that particular tissue. And then you have to draw the labeled diagrams. Diagrams without labeling are of no use. So a diagram should be very simple, drawn in pencil, and the labeling should be there, whatever the identifying features are there. So this was about the aim. Then the materials required. You will require permanent slides of parenchyma tissue, sclerenchyma tissue, striated muscle fiber, nerve cell, and the compound microscope. Now we have studied the compound microscope and its parts, so you are a little familiar with it. I know it does not compensate from going into the actual lab and doing it, but let us be hopeful. Very soon we will be back in our actual labs and you will be able to handle these microscopes. Then comes the procedure. Now here the steps for procedure in the textbooks, etc., they are written like they are being ordering. Do this, do that. But when you write it in your textbook, you have to write it as if you have actually done it. So, for example, you will write, placed the compound microscope where proper light could be received and reflected on the slide. So, you have to change it as if you are doing it yourself. The second point is, place the permanent slides one by one and observe the structure and drew the diagrams. So these are the first three steps: aims, material required, procedure. Now it is not like other experiments where you are performing. So simple procedure, you just have to observe. Then the next step would be observations, where you will be writing down what you are seeing for the slide. And at the end there would be some precautions. So that is how you will be writing the practical. A very quick review: plant tissues, so many different types. I will not go through them. Here in this, we have to do only the simple permanent tissues, parenchyma and sclerenchyma. But I thought, to avoid confusion, very quickly I'll be showing you the other 2 or 3 tissues also. So let us see. The first is the parenchyma tissue. Quickly, I would like you to recollect what you have studied in the video on plant tissues. Here, for your reference, this is the simple living cells having very thin cell walls. They are loosely packed and you can see they have lot of intercellular spaces. Now because they have thin cell walls, you will find them wherever there are soft parts in the plant. And they also form what is called the basic packaging tissue. See, inside the plant there will be definitely tissues which will be carrying food, there will be definitely tissues which will be transporting water, and other which will be providing strength to the plant. So wherever there are empty spaces between these tissue, you will find parenchyma filling up those places. So that is what it means, forms the basic packaging tissue. If it is filling up the spaces, definitely it is providing some support to the plant. And if it is phloem, then it will also be storing food, say for example in potato, etc., where starch is stored. And if it is like stem and roots, it will also be helping in the movement of water. And it has got 2 modifications, if you remember: chlorenchyma, where the parenchyma is packed with lot of chlorophyll and it is involved in photosynthesis. And then there is parenchyma, in which there are a lot of intercellular spaces filled with air, and that provides buoyancy to the aquatic plants, that is, it helps them to float. Then we come to quickly the collenchyma. You don't have to draw this tissue, it is just for revision and for comparison. So if you see, these are also living cells, but they are elongated and they are irregularly thickened. That means only at certain points at the corners they have certain additional material, and that material is pectin, and it is normally filling up all the intercellular spaces, so there are hardly any intercellular spaces. Now, from the structure that it has got some material deposited, what does it mean? That the cell wall has become thickened at certain places, and definitely it will be providing support. So it is, collenchyma is found in the leaf stalks just below the epidermis. If you feel the leaf stalk, it is little hard. And the function is, it allows easy bending or flexibility, or we can say elastic strength, or we can say tensile strength in the various parts of the plant wherever it is present without breaking. Now, what does this mean without breaking? You may have observed that if there is a storm and there are strong winds, then the tall trees, they will bend and they will be broken, they will be uprooted. But these small plants, they simply bend with the wind and bounce back to their original position. And it also, again, in small plants, it will providing that strength to stand. So it provides the mechanical strength also. Then we come to sclerenchyma. Very important, it's made up of dead cells. Here you can also get in your theory that you differentiate between parenchyma and sclerenchyma. So first point would be they are living cells, and here they are dead cells. And they are of two types. Number one, the fibers, which are long and narrow. So on the left hand side you can see how they are long and they are very narrow. And on the right hand side is the transverse section. And what is clear in the transverse section is that the cell walls are very thick because of a material called lignin. Now, when the cell wall becomes thick, what is happening? The inside space is very less. Anyway, it is dead cells. Inside space is very less. And from here only we can know that it is for providing strength. So, it provides mechanical strength to plant parts, making them hard and stiff. For example, the husk of coconut. Then the second are the stone cells or sclerites, and they are so thick, the wall is so thick that there's hardly any space in the center, and you find them in the hard covering of seeds and nuts like walnuts, akhroat, and in the pulp of guava and pears. When you eat the pear, especially nashpati, in your mouth you can feel as if you are eating some sand particles, and those are nothing but the sclerete cells. Then quickly the xylem. This again you don't have to draw, but just for your revision, transport of water and minerals from soil to aerial parts, and they are made up of four types of cells. The tracheates, which are elongated, narrow, dead cells, and they have pits. Then the vessels, which are of larger diameter, and they have also have got thickenings but in different patterns. Then the xylem parenchyma and the xylem sclerenchyma. That is the parenchyma and the sclerenchyma, which is associated with this bundle. Then quickly the phloem. Again, the function, it transports food from where? The leaves where it is prepared to all the other parts where it will be utilized, and also from storage organs to the other parts. So whenever the plant is not photosynthesizing, then the food can be transported from the storage organs to the other parts, and it is made up of 4 types of cells, what you call the sieve tube cells, the companion cells, the phloem parenchyma, and the phloem sclerenchyma. Here is how it looks under the microscope. You can see the sieve tube cells, they are also dead cells, and they have got at the cross walls these sieve plates, which you can see very clearly in this transfer section. This, it looks like chalni. So from here the name sieve tube cell arises. And then the companion cells you can see with the nucleus. Then we come quickly to the animal tissues. Here we have to do only the 2 animal tissues, nervous tissue and the muscular tissue. So first, nerve cells or neurons. This is how the tissue will look under the microscope. Now you are very familiar with this diagram, which is there in your textbook. So you have to draw this diagram in your book, and you have to draw the labels, and you have to write the characteristic features. That is, this tissue is made up of nerve cells or neurons, and then you have to describe the different parts like axon, which is the longest part of the nerve cell, then the dendrites, which are small branches, and you can also write about cyton or cell body, and the nerve endings. And where is it found? It is found in the brain, spinal cord and nerves. And functions include, it passes or it transmits signals from the external environment to the different body parts very quickly so that the muscle can give response. Then we have the skeletal muscle fiber. Here again you have seen this in the animal tissue also, the same slide. Easy to identify. This is what you have to draw in your practical copy. And what will you write? Where is this tissue located? It is found along with the bones, and it helps in the movement by contraction and relaxation. Now what are the structural characteristics? That these are unbranched cylindrical fibers. They are multinucleate. You can see different nuclei are present, and they show striations. Then they are voluntary, and they get tired or they are fatigued. These are very important 5 points you have to write along with the well labeled diagram. Then the next two the next 2 fibers are just for revision. The visceral muscle fiber, this is how it looks under the microscope, very tightly packed, spindle shape. You have to draw this in your, no, you don't draw this in your practical, you just learn it up from the theory point of view. So what are the characteristics where it is found? In the wall of organs like stomach, intestine, and these are having spindle shape. They are uninucleate, they are unstriated, they are involuntary, and they never fatigue. And finally, it is the cardiac muscle fiber. This is how it looks under the microscope. This is how it needs to be drawn in your theory. It is found in the wall of the heart only, nowhere else. So these are branched cylindrical fibers. They are uninucleate, they are striated, they are involuntary, and they never fatigue. So with this, children, we come to the practical part also. So in the end, you can write some 1 or 2 precautions which you have to take, that you handle the microscope carefully. You, after you have seen, you will put it away, all the things. All these are precautions that you must write, 1 or 2. And here, these are some references for you, which I have also referred, and you can also refer in preparing this video. So, thank you. Enjoy your learning. Be safe, be happy, and all the best.